Don, I want to make sure that we get to vaccines too. Do you want
Sure.
to just quickly summarize that for 30 seconds, and then we can move on to vaccines?
Yeah. So I just want to, from the start, is really that there are efforts, including the Therapeutics Accelerator, where we're screening all drugs that are known to be safe in humans, and seeing in a dish whether or not they're effective against coronavirus. That is not equivalent to being safe or effective, not equivalent to being effective against coronavirus in a person. There are bioavailability questions. There are questions about dosages and whether or not they're realistic. And also the fact that we need to know in a trial setting whether or not it is actually effective against the portion of illness that is attributable to the virus.
Yeah, and then the advantage that testing existing compounds has is that they've already gone through trials for safety. So you know you're starting with something that will be safe if it is found to also be effective. So now let's, we only have a few minutes left. And the vaccine development, I think, is one of the most interesting long-term questions here. So maybe just give a little bit of background on what's going to be necessary to get a vaccine. What are the different strategies? I mean, you hear people focused on protein-based vaccines, RNA-based vaccines. What are those different kinds of things? So what are the different kinds of approaches? Then again, realistically, what are the timeframes that we're looking at here for having something that can be widely deployed?
Yeah, so there are many different formats for inducing an immune response. You can try injecting directly into the muscle RNA or DNA that encodes viral proteins. Though the advantage of that approach is that you can start right away. The minute you know the genomic sequence, you can synthesize the gene in question and inject that right into the arm. So that's why those are the vaccines you're hearing the most conversation about when Dr. Fauci says on TV that these might be deployable in a year to 18 months. He's talking about the nucleic acid-based vaccines principally, which are already off to such a quick start because once you know the sequence, you just synthesize the gene and inject it into the arm. Now, the good news is that approach is fast. The caveat here is that in influenza, where we have some experience with this, nucleic acid vaccines, DNA-based vaccines work very well in mice, but haven't worked so well in larger animals and humans. And we're not entirely sure why that is. They still got a shot here. Every infection is different. They might work here, and you absolutely need to try it. But I think we can't rely on that just because it's quick doesn't mean it should be our only approach. I think this is a case where you want to be shooting every arrow in the quiver. That means protein-based vaccines. It means expressing viral proteins and looking at their ability to induce an immune response. You have to choose which protein you want to use or which collection of proteins you want to use. I think in this case, we mostly want to use the surface protein of the virus because we know that's the thing that binds to the receptor, and that binding to the receptor is imperative for infection, and that's an event that happens outside the cell and therefore can be blocked by an antibody. So spike glycoprotein or S protein, that's key. And you can do that by either expressing S protein and purifying the protein in recombinant yeast or higher cells, and then injecting the pure protein into people. You could try growing up whole virus particles and inactivating them with chemicals or heat or detergent, and using the disrupted virus particle itself. It's no longer infectious to induce. That has the advantage of not making any guesses about what the optimal antigen would be, but is of course much more complicated to do and requires biosafety and containment. But a killed virus like that would be analogous to the Salk vaccine for polio. And then there's other approaches where you take the viral gene for the surface protein and clone it into a harmless virus that has been used for other vaccines like adenovirus or certain forms of smallpox vaccine. And then what you're injecting into a person is the adenovirus that expresses that envelope protein. So these are all possible strategies for vaccination. And what I would say is, given the urgency of this situation, the government should be, and industry should be pursuing all of these lanes simultaneously. We don't have time to figure out, to make guesses in advance what's going to be the optimal approach. This is a very empiric subject. We need to test them all. And I'm very confident that that's going to happen. but we need to be sure that there's federal oversight of this and that every one of these approaches is being taken. Because even though some are faster than others, we really don't know what's going to be the most successful approach. And 18 months is the earliest any of these are going to come online. But I think, we've got to make a full court press. Every arrow from the quiver needs to be shot.
So are there approaches that you think are good ones that you're not seeing being tried right now by the major government?
I'm not privy to all the trials. I'm not privy to all the trials that are going on because, this effort is getting ramped up now. And it takes a while to make these recombinants, for example, or to express the proteins, get them in the appropriate state of purity, make sure they're immunogenic in animals before you start putting them into human beings. So, this isn't always public information until a clinical trial is set up. And so I'm assuming that because I have great faith in Dr. Fauci and I know him to be extremely well-informed immunologist, viral immunologist, I cannot imagine we are not doing all of these things, but I just think the public should know that this should be an all hands on deck effort. And I don't happen to know whether every single one of the vectors I just mentioned are being tested. I'd be surprised, however, if they were not.
So in terms of timing, you gave this time frame of potentially around 18 months for having this online.
At the early.
Can you give, so that's a little more conservative than some of the other estimates that we'd heard that it might be ready in the first half of next year. Is what you're talking about that the time frame for this being widely available in the U.S., for example, or are you talking about
deployable in the U.S.
up into billions of doses to be able to administer around the world?
Well, you can't ramp up something into the billions of doses range until you're certain that it is both safe and efficacious, right? And it's just not economically viable to do that for 16 different preparations. And so I think, to me, what I've heard Dr. Fauci say publicly is 12 to 18 months to deployment at the earliest. If the nucleic acid-based vaccines work, you're at the 12-month end of that range for deployment. Now, I don't know if he's talking about deployment globally or if he's talking about deployment in the U.S., because obviously the scale-up necessary for global distribution is quite profound. But there are also many other institutions that can participate in that scale-up. There are lots of biotech firms in India and China that specialize in the production of high-volume, low-margin products like vaccines. And I think that most vaccine manufacturers would be inclined to partner with a whole series of those enterprises in order to make the global distribution, possible, which is, it's what it's going to take. But I think that 12 to 18 month time frame, which I think is itself an optimistic one, is for American deployment.
And then what do you think, just typically from your time in industry, how long do you think it would take to be able to scale up, to be able to deliver this globally in billions of doses after that?
Yeah. I'm really not sure I can answer that question. By the time I was at Novartis got out of sold its vaccine business to another manufacturer. So I was never deeply involved in the manufacturing part of vaccine production. But it's a big endeavor to scale up. And my guess is that a variety of generic firms overseas would be taking that responsibility on in partnership with their American or European counterparts. I don't think any single company could probably meet the entire global demand. But these are things that I'd be speculating about. I wouldn't want to put a timeline on that. You can bet it would be a very high priority and banked fully by governments and charities as well as the private sector.
Yeah. So I want to be sensitive to time because we've been going for about an hour. But before we wrap up, is there anything that we haven't gotten a chance to cover that you want to make sure that we cover, or anything that you want to reinforce?
No, I think we've pretty much hit all the high points.
All right.
I mean, we haven't talked much about what strategies, how you would deploy these to come out of an epidemic, but that's probably going to take a little bit of time. And But I think these are the tests that we need to have, and we need to understand how to use them in order to think about how to come out of a lockdown. What would a staged exit from a lockdown look like? Maybe we save that for another time.
Yeah, I think that'll be one of the next of the next lives that we do. We'll probably be on that topic. Anyway, Don, thank you so much for joining us. This has been really interesting, and we covered a lot of ground. So thank you not only for joining us today and helping to explain some of these ideas about what needs to happen to people broadly, but I also appreciate the work that you're doing as an important advisor to the BioHub and helping with the efforts there. So thank you for everything that you do. It's great to see you.
Thanks,
Stay
Don.
well.
Stay well.
Thank you very much. My pleasure. Thanks for having me.
For everyone on the stream, I hope you're all staying well, staying healthy and safe, and we will see you all soon.